Hello friends, very good evening to all of you. In this particular video, we are going to talk about the mechanism of muscle contraction and relaxation also. So both the things we will look here. So first of all, I am drawing a structure of neuron for you. Okay. I hope it is visible. Let me draw here. Okay. So this is a neuron, which is motor neuron. We all know there are mainly two types of neuron, motor neuron and sensory neurons. Motor neurons bring signal from your brain to your body organs or body tissue. So this is motor neuron. It is going to pass the signal from brain to towards your muscles, the skeletal muscles. So here I am drawing a muscle fiber, which is a muscle cell. Okay, this is a muscle cell. This is the plasma membrane of the muscle cell, sarcolemma. So the neuron along with the muscle cell is called as the motor unit. This is called as motor unit. This is here I will write motor unit. Okay, neuron, motor neuron along with the muscle fiber. This is muscle fiber. Muscle cell, both togetherly called motor unit. Then there is a this sarcolemma and the end of the neuron, so axon terminal, collectively called as the neuromuscular junction. Neuromuscular junction, or there is another name called motor end plate. Motor end plate, the end of the neuron and the plasma membrane, sarcolemma of the muscular cell is collectively called neuromuscular junction, where neuron and muscles is having connection. That is neuromuscular junction or motor end plate. Then the mechanism of muscle contraction is explained with the sliding filament theory, which states that the thin filaments are going to slide over the thick filament. Suppose this is a thick filament and this is thin filament. So thin filaments are going to slide over the thick filament. So again I am drawing, suppose these two are Z lines, and we know that Z lines, the distance between two successive Z line, this is sarcomere. Sarcomere, the unit for contraction. Unit for contraction. And Z lines are firmly bound with thin filaments like this. These thin filaments are firmly bound by Z line like this. And there are thick filaments over here. There are thick filaments. So these thin filaments are going to slide over thick filament, and because they are firmly attached to Z lines, Z line is going to pulled inwards towards A band. This is A band. This is A band. So whenever thin filaments are sliding, they are going to pull Z line inwards towards A band. This is A band, right? And which result the contraction of sarcomere and ultimately contraction of muscle cells, because these filaments are attached with the muscle cells. So ultimately muscle will get contract, muscle cell will get contract, and whole muscle will contract. How it is happening? The motor neuron sends signals to this muscle to get contracted with the help of neurotransmitters. These are neurotransmitter, and especially acetylcholine. Acetylcholine is a neurotransmitter which is sent to the neuromuscular junction where there are receptors of this acetylcholine. So as these receptor receives the acetylcholine from motor neuron, what will happen? It will result the action potential in the sarcolemma. And there are some Na plus ions present outside your sarcolemma. Those Na plus ions will come into the sarcoplasm. And by looking these Na plus ions, who is a friend of Na plus? Calcium. So we all know that there are sarcoplasmic reticulum who has calcium in it, Ca++. So these calcium ions will come into the sarcoplasm, Ca++. So these calcium ions, we all know that they are having binding capacity to bind the troponin which is present in the actin thin filament. So these calcium are going to bind troponin. Suppose this is troponin, and this calcium will bind to troponin, which is going to remove this troponin from the myosin binding site. Okay, and this troponin will be removed, which results the unmasking of myosin binding site, so that myosin head present on the thick filament will bind to the thin filament like this. Here I will draw. I hope it is clear. See, there are thin filaments made up of tropomyosin and F-actin, right? So there are sites present which is masked by troponin. So this troponin is removed so that these sites are open to bind the myosin filament, myosin head. So because of the removal of troponin, which is attached with calcium, so it is gone from this site. Because of the removal of troponin, this myosin head has binding capacity to actin filament. So this is going to bind over there. And we all know that myosin head has the ATP binding site. So with the help of ATP and hydrolyzing that ATP, the energy from that ATP is going to help this myosin head to bind to actin filament. And this head has a unique capacity to rotate its head like this. So it is bound to actin filament and then it will rotate. So it is ultimately pulling this actin, which means thin filament, inwards. So this is going to happen here. So because this head is rotating like this, it is going to pull these thin filaments and ultimately it is going to pull the Z line inwards. Again next ATP will bind and cross bridge will be broken, and then again that ATP will be hydrolyzed and cross bridge will be formed, and then again rotating. So this helps to slide this thin filament over thick filament, and ultimately this Z line will come inwards towards A band, which results in the shortening of sarcomere, which is the unit for contraction. So sarcomere will be short and thus it is contraction of the sarcomere of the muscle cell of the whole muscle. Then how this process of sliding takes place till calcium ions are pumped back into the cisternae, sarcoplasmic cisternae or sarcoplasmic reticulum. These calcium ions will again pump back into the sarcoplasmic reticulum, and then it will again, this myosin binding site will be covered with the help of troponin, and this sliding will stop. It will result these Z lines to its original position. This is relaxation. So I hope it is clear. Just imagine like there is a train which is muscle fiber. And there are so many bogies in it, right? And the contraction of one bogie will help to contract all the bogies and resulting in shortening of the whole train. Just like that, in the muscle fiber, there are so many myofibril. Myofibril has so many sarcomeres, and sarcomere has are getting contracted, ultimately resulting in the contraction of whole muscle fiber and whole muscle in your body. So this is going to happen. I hope it is clear now how muscle contraction is getting, uh, uh, how muscle contraction process is working, and the relaxation is going in inside your body. So this is about it, guys. I hope it is clear now. If you like this video, if you found this video helpful, please hit the like button and share it with your friends so that they can also get benefit from it. Thank you so much.